So I'm Peter Littlewood, I'm the Argonne Lab Director, and you'll be hearing more, not so much from me, but about me later. So I actually have two things to do. So first thing I need to do is I need to point out where the exits are in the case that we need to do so. They're at the back, but I recommend if you can, you would come out this way and go out into the parking lot. I'm not expecting that we'll have to evacuate, but I'd just like to remind you where to look. The next thing I have to do actually is to introduce you to my former boss, or maybe still my current boss, Eric Isaacs, who's Provost of the University of Chicago and was until not so long ago Director of Argonne National Labs. So some of you will have seen him standing up here before now. And so let me hand over to Eric.
Thank you, Peter. And again, welcome to Argonne Out Loud. I feel a little funny standing up here since I haven't done so in a few months, but I'm really delighted to be here tonight to introduce our speaker. I do want to say that Argonne National Labs, and many of you know it, and some of you even work here, is really one of the most outstanding national labs in the country, doing some of the most exciting and interesting science, and you'll hear about some of that tonight. When Jack first invited me and Peter to participate in tonight's Argonne Out Loud lecture, I was actually quite surprised by the form of his request, and you'll see what I mean momentarily. But he is doing really great science, and I am an experimental physicist, so in my opinion, there's almost nothing I wouldn't do in the name of great science, and I think you'll see what that means momentarily. So let me say a few words about Jack. He's really remarkable individual. Tonight, as you see, he'll be talking about Invisible Influence, a Bacterial Guide to Your Health, which will feature Jack. And it'll also feature, in some way, Peter and I, which we have yet to understand what that means. So Jack is a microbial ecologist here at Argonne National Labs. He's a superb scientist here, but he's also a professor at the University of Chicago in the Department of Ecology and Evolution, and he's just all over the place. His research is focused on exploring microbial communities, how they assemble themselves, how they evolve, and how they interact with the world around them, with a particular interest on how those communities affect us as human beings. And you'll see that tonight. He's also, in addition to being a great scientist, both theoretically, experimentally, and computationally, he also is getting a lot of public attention these days. You might have even read about him or seen him. He's recently been covered in Time. He's been covered by Newsweek, National Geographic, and the Washington Post. And if that's not enough, he was just last week, recognized by Crain's Chicago Business as the best of 40 under 40, which we're always delighted to see in our earlier career scientist. And Jack clearly is well deserving of that kind of award. So before we bring Jack on stage, he would like to show us a brief video that will provide a little background to what microbial ecology is about, what he's going to be doing, what a microbial ecologist actually does, and it gives some themes, a sense of the themes that he'll be talking about tonight. So I'll step off stage, we'll watch a video, and then I'll be right back.
I'm Dr. Jack Gilbert, and you're covered in bacteria. Welcome to Chicago. There are 10 million people in the metropolitan area, and every single one of those people is shedding from their bodies 15 million microbial cells an hour. That means that every time you interact with someone in your day-to-day life, you're picking up microbial signatures from them, and that's shaping your microbiome and altering your life. And that's fundamental to our research, trying to understand that interaction, map it, structure it, and use it to understand health dynamics. So for the last 100 to 150 years, we've been trying to wipe out all microbes in our environment. With antibiotics and sterilization agents, we've been going at them hammer and tongs, trying to kill them all. And we're now starting to realize that those microbes are really important for our ecosystem and our health. They train our immune system to be strong and healthy. We need them in our environment. So you have 100 trillion bacterial cells inside your body. They outnumber your own cells 10 to 1. And what we're starting to understand is by looking at those organisms and finding out which ones you have versus which ones your friends have, we can say intricate things about your health and your well-being. For the last 10 years or so, we've been applying next-generation sequencing technologies like those we have at Argonne National Laboratory, which are like telescopes, helping us to see the microbial world inside our bodies and our environments in unprecedented detail. And we're starting to learn things about the microbial interaction we share with our environments. Our homes, our offices, our cities shape our microbial cells, and that in turn shapes our health. By looking at these things in detail, we're finding out that bacteria can maybe, if we change the ones inside you, cure things like autism, Alzheimer's, even cancers. We can look at obesity as an epidemic. We can understand neurological conditions like schizophrenia and reshape them. Allergies, asthma, they can be eradicated by adding the right kinds of bacteria back in. So it's fundamental that we understand this in a rich and cohesive way.
Welcome, please join me in welcoming Dr. Jack Gilbert.
Thank you very much. That was a very cold day, so if I looked a little bit peaky, it wasn't because I'm English, it was because the air was cold. There's my Twitter handle. Anyone that wants to help me increase my number of Twitter followers, you're going to be on Twitter right now. Let's take a photo of you. Use the photo, and I'll tweet you. There we are. So I'm going to give you an overview today of our current understanding, our, if you will, our best knowledge practice about the bacterial world, the viral world that lives inside you. As I said in the video just now, we've been trying to eradicate this world ever since we found out that it caused disease. So Louis Pasteur and Robert Koch in the 19th century really started to help us identify that the bacteria that were killing literally hundreds of millions of people a year around the world were bacteria, bacterial orientated, and viruses. And since then, we've been trying desperately to find ways to cure those diseases to great effect. Nowadays, we can basically enable millions of people, hundreds of millions of people to live who wouldn't have been able to live even 50 years ago. And this is fantastic. This is a wonderful advance in our understanding. But we've reached a tipping point. We've got to the position where we are eradicating so many bacteria in our world, our bodies are not responding in the way that they evolved to respond. You grew up in a world where you were constantly bombarded with infections, with parasites that lodged inside your colon, inside your body, for your entire life. And that affected your immune system. Your immune system developed, it evolved to understand the presence of those organisms. It understood that they were there. Whether they were healthy or, or harmful, the immune system evolved to deal with their presence. And when they're removed, the immune system gets confused. It's like, well, hell, where did these things go? I'm prepared, I'm ready, come on, bring them on, but they're not there anymore. And so you get attacks. You get the immune system starting to overreact and attack the body. So I'm going to give you some anecdotal evidence and some hardcore scientific evidence which shows that the bacteria in your body are mostly probably helpful to you. And that there's a transition state that we need to understand about when those helpful bacteria become harmful. So I'm going to start out by introducing you to the rest of yourself. You're only about 10 trillion human cells, give or take. You're 100 trillion bacterial cells, give or take. Let's say that the bacteria in your body, those 100 trillion cells, they weigh about 2 pounds, and that's a more meaningful number. 2 pounds of bacterial mass inside and on your body. That's about the same weight as your brain. So if you think your brain's important, well, the bacteria weigh the same, so maybe they're important too, right? Well, they colonize every surface in your body. Every surface that they can get hold of and get onto, they colonize. But they don't do this passively. Your body actively recruits bacteria. It releases antibodies into the gut, into your mouth, which are designed, their sole purpose is to recruit bacteria, to allow them to grow more effectively in that environment. So if you thought your body was trying to kill bacteria all the time, that's not true. It actively recruits them. And that's very important to our understanding. Why did the body evolve to actively recruit bacteria? For fundamental reasons. One of the best ones is if you have a bacterial army inside you that's benign and potentially non-infective, it can protect you against bacterial pathogens, against those invaders. So if you are already colonized, if all of your real estate is already populated, there can be no inadvertent colonization. We don't get the situation where loads of bad pathogens move in and take up a home and residence inside your body. So the body actively recruits those bacteria to prevent that. So your bacterial world is quite epic. It's a massive system, and we are only just in the last 10 or 15 years really starting to understand it in a real way, in a way that can have translational impact for medicine. And that's the most important. Taking this data, taking our understanding and knowledge that I'm going to talk to you about today, and translating that into something which can have real efficacy for treatment of diseases and conditions. So we need microbes. We've been, since about 1920, developing animal models. And when I say animal model, I just mean an animal, like a mouse or a rabbit or a guinea pig. We use those as models as an example of a human condition. One of the great ones is the mouse. And the mouse has been a brilliant animal model. These are mouse intestines, okay? We need bacteria to help our body, our physiology, develop in a normal way. This is the cecum. This is a part of the mouse gut that contains bacteria. This is a cecum from a germ-free mouse, a mouse that has never seen a bacterium before. This is the cecum from a mouse that's just been colonized by one particular bacterial species. And this is the cecum from a mouse that's conventionalized. What I mean by that is it's got a natural microbial flora of some 500 to 1,000 different bacterial species. When the mouse is colonized by its natural flora, it develops a normal physiology. Its body responds and works in the way that you would normally expect. Germ-free animals, animals that have never seen a bacterium, you'd think that they actually sometimes live longer than normal mice, but they live longer because they never see a parasite or a pathogen. So they never have any kind of burden on them. But they live in a very strange world. Their physiology is disturbed, it's mutated. Their neurology is even worse. They show very distinct differences in behavior, and I'll mention that. So we need bacteria to train our body, to make it develop and grow in the most certain, specific way, to make us, well, what you are. If you're a healthy person, you've been conventionalized in the right way. We know, excuse the lewd pornography of flies, we know that we can change behavior by changing bacteria, okay? In this instance, there's a female fly and a male fly doing what flies do. The bacterial population in the gut of one fly can be changed. I can take the microbes living in the intestine of one fly and transplant them into another fly, and I can make that second fly want to mate with the fly that the first fly wanted to mate with. Get the details right, but you can transfer a behavioral phenotype. You can make a mate selection. You can make who this fly wants to spend the rest of its fly life with change purely by changing the bacteria. The bacteria interact with the immune system of the host, changing certain neurological impulses between the gut and the brain, which alters behavior. So if you change the bacteria, you change neurological behavior. And we can do this routinely. We can also do it in mammals. So again, in a mouse model, this is a great model where we look at the microbes in the gut of the mouse, and we see how they influence brain activity, okay? So we do this in many ways. We try desperately as scientists to find ways that we can compare the behavior of these mice to human behavior. So we tend to end up anthropomorphizing the mice. We give them human qualities they simply don't have. So I'm going to do that right here. I'm going to say we have two examples of a mouse. We have a brave mouse and an anxious mouse. And this is called an elevated maze scenario, okay? It's not a very big maze. It has basically just 4 options. We put the mouse in the middle of the maze. We stick him right there between, in the middle of these 4 things. It's about ½ a meter off the ground, okay? And mice don't like being in elevated spaces if they are feeling anxious. So an anxious mouse tends to run inside the box and stay there, whereas a brave mouse tends to explore the platform in a more detailed way. We've shown that mice that are brave have different microbial signatures to mice that are anxious. In fact, some of the bravest mice are actually germ-free mice. They have a neurological tendency towards suicide. They go a little bit crazy and they'll do anything. They'll run up to the edge and jump off. But if we have a certain microbial community in there, they're brave, they'll explore their environment, they have a lowered anxiety. And we can transfer the microbes from an anxious mouse into a brave mouse and make that brave mouse anxious, and vice versa. We can take an anxious mouse and make it brave again. And this has big implications for human behavior. Depression and anxiety are crippling problems in our society today, across many nations, not just the United States, but Europe and Asia as well. We have fundamentally failed as a society to really handle this in a strong way. If we can understand the mechanism by which depression and anxiety affect our population, we can determine new ways to actually deal with those conditions. Quite possibly, the microbiome is an answer to that, or at least one of the answers. In our society, we have an Overselling the Microbiome award. So if it seems like I'm trying to say that the microbiome can cure all ills, I'll only tell the funding agencies that. But for you, good public, it's not necessarily true, but we are starting to understand the intricate necessity of the microbiome in our health. We are an ecosystem. We do not exist as just human cells in a void. We exist as an ecosystem of organisms, and we have to understand that, accept it, and use that information to fundamentally change our health policies. We know that we can change whether an animal model has a food allergy by changing the bacteria. So obviously food allergies are another crippling condition, especially affecting the youth of developed countries. They're very, very rare in developing countries, but very, very prevalent in developed countries. And there's a hypothesis called the hygiene hypothesis, which stipulates that children are developing these autoimmune conditions because they're not exposed to enough biodiversity when they're growing up. When their bodies and their immune systems are developing, they're not getting enough exposure, not enough training. We've tended to show that that is the case in mice models. So in this situation, we've developed a mouse which has crippling peanut allergies. It's amazing what we do to mice. It's quite horrific. I apologize if anyone's squeamish about that. But if you expose this mouse to peanuts, it can die. It's very severe. If we see this graph basically shows the amount of response that mouse shows to the peanut. If it's a very elevated response, it's a bad thing. That mouse will die. So here we have a germ-free mouse, this GF. And the germ-free mouse has a massive over-elevated response to that peanut, okay? Very negative, and it can kill it. If we add in this Clostridium bacteria, this one organism, it significantly reduces the mouse's response to the peanut. And so colleagues, including Dion Antonopoulos, who works here at Argonne, and Kathy Nagler at the University of Chicago, have been developing strategies to use this information to develop programs that we could maybe take to clinical trials. We want to start trying if we can add these bacteria into children at an early age, maybe it can prevent these allergies from developing. We also know that bacteria can affect your senses. So work we've been doing with Italian groups, this is a wine study, so of course Italian groups, has explored the fact that there's a link between how much you sense food or drink and your microbiome. So the bacteria in our oral cavity are influence our health, our obesity, many other things. And I'll talk about obesity in a minute. I'll give you a detailed description of that. But in this system, we looked at the bacteria living in the oral cavity of obese men and normal weight men. And what we saw was that obese men, the microbes were generating less aromatic compounds from the wine. Subsequently, the men couldn't sense the wine as much. And we hypothesize that this means that they are satiated. They're less satisfied by a small amount of wine. In order to be satisfied, in order to have a satiated response, they need to consume more wine. And wine's not an obese-causing phenotype, but it leads to other foods. And we believe the bacteria in the saliva of these obese men is leading to this initiative. What causes them to have different bacteria? Potentially different diets, and I'll show that in a minute. We know that bacteria can affect neurological development in fundamental ways. In fact, we can cure autism in a mouse by altering its microbiota. So we can create an autism mouse patient. Again, this is an anthropomorphization of a mouse. But we can infect a mother mouse, a pregnant female mouse, with a virus particle that basically leads to a pup being born with a leaky gut syndrome and neurological behavior very reminiscent of autistic children. I have an autistic son. He's 7 years old, and he's perfectly capable of surviving in this world, but I'm very interested in trying to see if I can help his development as he grows older. So we've been working with groups out at Stanford and out at Caltech and exploring ways in which we can change the microbiome and affect a real difference in the physiology of these mice, potentially with the way of moving forward to a clinical trial whereby we could alter something about the lifestyle of the host, of the human, and affect a change in their behavior and their physiology. In this mouse model, that had this leaky gut, so its bacteria in its intestine pumped loads of metabolites into its bloodstream. It also affected neurology by passing the wrong kinds of signals from the gut to the brain and causing stereotypical behaviors and antisocial behaviors in the mice. We altered, this group out in Caltech under Sarkis Mazmanian and Paul Patterson looked at affecting antibiotics, the effect of different foods, different diets, and the effects of probiotics. And they had one particular probiotic, a bifidobacteria. And this bifidobacteria, when they added it in, it virtually ameliorated all of the conditions, the physiological leaky gut and the neurological behavioral distortion that this mouse suffered from. Again, it's only a mouse. Again, it's only a model. But it really helps us to identify particular traits and links between the bacterial community and the potential host response. We also know that it's another condition I'm very interested in. All of my grandparents, apart from one, suffer from Alzheimer's. My mother has a genetic signal factor for Alzheimer's. I have that same signal factor, and so does my eldest son. So it might be early onset, I'm not sure. But we're very interested in what triggers the onset of Alzheimer's. We know there are genetically predisposing factors, but it can happen at different phases in life, and there doesn't appear to be a genetic signature that's robust enough to determine that. Not in our genome. But we know that bacteria can produce compounds which are heavily correlated with onset. So if there are these compounds, things like beta-N-methylamino-L-alanine, also called BNMLA, or brain-derived neurotrophic factors, or what we call gamma, so gamma-aminobutyric acid. If we find these signals in the serum, in the blood of a patient, it's quite likely they are in the progress of having an onset of Alzheimer's development. And this leads to neurological regression and a breakdown in neurological pathways. And the bacteria, all of these compounds, are microbially mediated. So if we put these kinds of bacteria, not the right ones, but put the wrong kinds of bacteria in a mouse, they can mediate the levels of these chemicals, and in the mouse models we've seen, lead to the onset of an Alzheimer's neurological trait. And this is key. Most of the bacteria that produce and mediate the levels of these chemicals can be altered by diet. So if we can figure out how to treat people with a specific diet, and that might mean doing away with steaks or maybe even granola. We don't know what diet it is yet. It's very hard to do these experiments in humans. But if we can find the right diet, we can potentially lead to a significant reduction in the onset of Alzheimer's. And in aging populations such as we have in the West, this is a significant concern. Obesity is an interesting one. Since we really uncovered bacteria as having an influence upon our physiology, we've been very interested in obesity. One of the reasons is it's a major problem in Western societies, in developing societies as well, to a certain extent. It appears to have some correlation with genetics, but it's a very poor correlation. In fact, genetics really only predicts obesity about 50-50. So about 50% of the time, genetics is accurate at predicting whether you're obese or not. And that's a yes or no response, so it's not particularly useful. We know that environment plays a role. Obviously, there are multiple factors, but diet is a significant one. But we've been trying to understand the role that diet plays, the mechanism by which diet leads to obesity. So this is a great study produced by a Chinese group out in one of our collaborators out of Shanghai. It was the actual collaborator himself who was morbidly obese, so he had a vested interest in this research. And he put himself on a whole grain diet, so he only ate whole grain food for about 6 months. And he was 385 pounds, he lost 113 pounds. And he measured his bacterial community in his gut via his stool, which is a proxy for the bacteria living in your gut, and we'll talk about that later, guys. So he looked at the microbiome in his stool, and from before he lost weight to after he lost weight, he saw one particular bacterial species which became virtually absent towards the end. It disappeared from his ecosystem inside his body. And if you lose an organism from your ecosystem, it's going to have an effect. If you lost the lions from the savannah, it's going to have an effect. And so he was interested to know what this bacterium did. It's called Enterobacter cloacae B29, if you're interested. It looks something like this. And he did some fascinating experiments. He took this bacteria and he put it into another mouse model. And mice that contained this bacteria in their gut put on about 100% more weight than mice without this bacteria, eating exactly the same food. So they could eat 2,000 calories a day, and if you had this bacteria in your gut, you're going to put on a lot more weight than without this bacteria. And what's interesting, the obvious response from the scientific community when we first saw this evidence was, well, the bacteria's making the food more bioavailable to the host, so the host's getting more energy and putting on more weight, right? But we don't think that's true, and I'll show you why in a minute. Another example of an obese-causing phenotype is antibiotics. We've known for many years that we can treat animals with antibiotics when they're growing up and they gain more weight. This, in fact, leads to 80% of antibiotics generated in the US being used in the animal food industry. If you want a slightly plumper chicken or a slightly plumper pig, you've got to pump antibiotics into it. We're trying to overturn that. We're trying to find better ways of increasing animal mass. I don't think antibiotics is necessarily an ideal solution. But we also show that the same is true in animal models of human behavior, of human physiology. So if we give a mouse a low dose of penicillin when it's a pup, it will change its microbiome significantly. And that microbiome leads to the mouse gaining weight. That's correlative. You change the microbiome, the mouse gains weight. Yeah, it happens every time, but it doesn't mean anything. If we take the microbiome from that now obese mouse and put it into one of our germ-free animals, these animals which have never experienced a bacterium, it also puts on weight. So the microbiome transfers that obese phenotype. And this is fundamental. My child had antibiotics shortly after birth. Most C-sections and a lot of vaginal deliveries now are predicated on the use of antibiotics during delivery to preempt any infections. A lot of children get antibiotics throughout their early life for earaches and sore throats. Most of these are probably unnecessary due to viral infections which can't be treated with antibiotics. So we're starting to understand that potentially some of the conditions or ailments that we suffer in our modern society may be linked to the way in which we medicate our children. And there's a lot of evidence to support this. This is no longer circumstantial evidence. It's becoming a lot more rigorous. And I foresee a future whereby lawyers drive everything. I've always said this. If you If you want to change something, you've got to get somebody to sue somebody for something. So I see a future where somebody will take some of this evidence, maybe some more concrete evidence, and sue their doctor for giving their child antibiotics early in life. And if that happens, and they're successful, then doctors are going to become very paranoid about giving antibiotics early in life. And that makes me nervous. Hopefully it will have a beneficial effect, but we also don't want to get into a situation where we're not taking antibiotics and therefore we get outbreaks of disease. Antibiotics are a very, very powerful and useful tool, very important for the health and safety of our population, but we need to learn how to use them responsibly. And that's hopefully part and parcel of what the evidence we're generating now will tell us. This is a paper that just came out, but it's a fascinating paper, and I had to include it. Twins. For years, we looked at twins, and we looked at normal siblings, monozygotic twins who are genetically identical, come from the same egg, and dizygotic twins who come from different eggs but are raised in the same womb. And we couldn't see any significant effect of genetics, i.e. the monozygotic twins who came from the same egg didn't appear to have a different microbiome from dizygotic twins. This study looked at hundreds of pairs of twins and examined the difference in their microbiota. And they found a very, very small but significant effect of the host genetics on the microbiome. So your genetics can play a role, but it's a very, very small role. But it's a fascinating part of this study in the fact that they found that in monozygotic twins and dizygotic twins who were all of a low body mass index, they were all a healthy weight, they had one particular microbe that they all shared. And they were able to culture this microbe. It's called Christianella minuta. They were able to culture that microbe, and they put it into mice, again, like the previous study. The previous study, the Enterobacter cloacae B29, when we added that, the mouse gained weight. When we add this Christianella minuta, the mouse lose weight. We can give it to an obese mouse, and the mouse will lose weight. Again, I have several of my friends who, when they saw this result, said, "Can you get me this bacteria? Can you get it for me now?" It'd be great. Put it in a probiotic, drink it, and you can eat all the hamburgers you want. Maybe. Or probably not. That's the risk we run from finding this data and then it running riot without us having the proper evidence in place to be able to relay this information appropriately to the public. But we do know that this is a critical step in our understanding of this relationship. So we've been working at the University of Chicago to try and understand the mechanisms by which the microbiome affects the obese phenotype. And I said it's not necessarily the bacterial communities munching the food up more and making that energy available to the host. It's different effects. And so work with Eugene Chang at University of Chicago and Sean Gibbons and myself and Sarah Owens here at Argonne National Laboratory and Jared Hampton, Marcel, and a whole team of others have been examining the effect of high-fat diets on the microbiome. Okay? So when you eat a lot of high fat, it changes your bacterial community. I mean, that's not rocket science, that's a given. As my mother says whenever we publish a paper, she goes, "I've known that all my life. You just publishing this? What? I could have told you this." And I'm like, "Oh, well, yeah, but we have evidence now, Mum." And she's like, "Yeah, we have evidence. Should listen to your mother." Well, we have fantastic evidence that the bacteria influence the body in very specific ways. One, when you eat a lot of high fat, they produce a lot of metabolites, chemicals, from the bacteria which get into the bloodstream and they influence liver function. They change the pattern of gene expression in the liver. So, you know, your liver's made up of genomes, the genes are expressed, and when they change expression, it actually affects hormones in your body. So the hormones in your body change how much fat you put on. So if you have the wrong kind of bacteria producing the wrong kind of chemicals, they change your liver's capacity to produce fat cells. And we see this, that when you eat a high-fat diet, yeah, you're getting a bit more energy, but it can't explain all the weight you put on. But it changes the bacterial community, which generates these metabolites, which tricks the body into putting on a lot more fat. And I'll explain what I mean by tricked in a minute, but there's another link here. It's a positive feedback mechanism. The bacteria also relay and generate information at the gut lining. And this is relayed as a neurological impulse, a nerve impulse up into the brain, which releases certain more chemicals into the brain which stimulate behavior. Much the same way as with the mating flies and the brave and anxious mice, the gut bacteria influencing behavior. But in this instance, they're influencing appetite. But very specifically as well, the bacteria appear to influence appetite and cravings for the kinds of food which led to them becoming abundant in the first place. They are tricking the human being, and again I'm anthropomorphizing bacteria tremendously here, but they're tricking the human being into eating the kinds of foods which create the kind of ecosystem they love inside your gut. These guys love a high-fat diet. They love a high-fat ecosystem. So they trick your brain into having cravings for high-fat food. And they also make you gain more weight, so you become more sedentary. And so you get into this positive feedback mechanism by which you crave more fatty foods, you eat more fatty foods, you put on more weight, and ad infinitum, and you become morbidly obese. We see a similar response neurologically and in the liver in people who have chronic fatigue, people who work night shifts or who fly too much. I do 150,000 miles a year of traveling for Peter. He's a taskmaster. So it's fly here, talk about this, do this, jump. I'm just looking to get fired tonight. And the microbiome of people that have this chronic fatigue, that have chronic jet lag, or they work a night shift, they can gain more weight for the same calorific intake as somebody who's getting plenty of good sleep. Okay? And so that system is the same mechanism, but when you eat a high-fat diet, your body responds in the same way. So a high-fat diet can lead to your body being tricked to think it's not getting enough sleep. And so it enters into this sedentary lifestyle of putting on more weight and craving more fatty foods. And so we're just starting to understand this mechanism. And if we can understand it, we can manipulate it. And that's hopefully the goal of the research, is to try and understand that. So we're using some of the high-performance computers here, and we have some of the fastest computers on Earth on this campus, to really model this relationship. And so you may have seen a great display out there by Peter Larson, who's working with mathematical modeling and metabolomics. And we also have Chris Henry here at Argonne, who's developing chemical models. And combining those techniques, we can start to create this system in a computer. That's hopefully better. We won't be able to, we can do away with using mice, and we can just do this in a computer. We're not quite there yet. It's a lot more complicated than people think, but hopefully in the future we'll get to a situation where we can understand the best strategy for teaching or treating a person who has obesity based on their lifestyle, their traits, their diet, and understand how to affect it. I'm interested in where microbes come from. I'm an ecologist. I also like Calvin and Hobbes. But I'm an ecologist, and I'm fundamentally interested in the origin of bacteria. Where did that morbidly obese guy from China get his Enterobacter cloaceae B29? Where did it turn up from? Did he get it from his mother? Did he pick it up from another person that touched him, or a food he ate, or some water he drank? Where did he collect this bacterium from? So we're interested in understanding that relationship. We know that the bacterial communities inside our body mostly come from our mother to start with. So your mode of birth affects your microbiome. And if you're born via C-section, which is something we haven't evolved with, you actually start out with a skin bacterial community colonizing your body. Obviously, that's totally opposite from the standard mode of delivery, where you get the standard mode of microbial colonization. And we're just starting to understand this. So a baby born shortly after a vaginal delivery looks like the mother's vaginal microbiota. A baby born via cesarean looks like the mother's skin. And we don't know what effect this has. It gets even more complicated. Breast milk that the baby feeds on has its own microbiome, very complex microbiome with some 100 to 700 different species in there. The mother actively recruits that bacterial community from her gut up through her lymph system into the breast duct, forming a complex probiotic which she feeds to the baby. She's evolved to do this. This is a standard practice in the evolution of mammals. pumping bacteria into the child, priming their ecosystem, priming their immune system, priming their health. And we're just starting to understand what impact that could have. You take a baby born by a cesarean section and a mother who can't breastfeed, so she has to give a sterile formula feed, and you remove two major sources of colonization immediately. You then pump that kid full of antibiotics, what have you got? Massive dysbiosis. We have a lot of evidence, circumstantial correlative evidence, to suggest this is having an influence upon our population. We're not allowed to experiment on humans. Maybe I don't wish we could, but it would be so much easier if we just had a... Anyway, I won't go there. Peter won't allow me to do that. Bad Jack. But we're very interested in how we can experimentally determine the influence that this could have upon human development. If we could maybe discover the right kinds of bacteria and add them into that formula feed, maybe we could do away with the necessity for breastfeeding. Not do away with it, it's a fantastic need for it, but if you can't breastfeed, maybe we can recreate it in a bottle. Maybe we can colonize a child early in birth with the microbes that they need to start with. And if we can do that, we can have a real impact on our life. But as soon as that baby is released into the ecosystem, into the world, into the environment you're sitting in right now, it experiences other colonization events. The bacteria in the world around you, and you are surrounded by bacteria right now. Each one of you, as I said in my video, is releasing about 15 million bacterial cells into this room an hour. Most of them coming from your skin. You in the front are getting a nice dose of my oral microbiota. Apologies. It's probably a good one, I don't know. And we're just starting to understand this microbial universe. So each of you is microbially unique, because none of you have the same kind of microbial exposures as another person. It's virtually impossible for that to happen. Even if you're identical twins, as I said, you're only slightly more microbiologically similar than normal twins. Even identical twins live slightly different lives. One might get kissed by a dog and the other one might not get kissed by a dog. One might dig in the garden, the other one might not. One might grow up living outside more, the other one might be inside more. And we're starting to understand how that affects and changes our life. So we wanted to go inside buildings. We wanted to look inside buildings and understand the microbiome of those buildings. Best place to start is your home. Your home is where you spend a lot of your time. If you're an infant below the age of 1, you spend 99% of your time in the home, on average in our Western society. That's a huge amount of time. And so we did the home microbiome project in order to try and understand that ecosystem and see, or literally create a map of the microbial movement between us and that ecosystem. So we could potentially maybe manipulate it in certain ways, create a better home. We found some interesting things that we can forensically identify people based on their microbiota in the home. The red lines in here, these are kitchen light switches on top and 7 different families. Kitchen light switches, front doorknobs, bathroom doorknobs, kitchen floors, bedroom floors. If it's more red, it meant we could identify the family by the microbiome on that surface. And so family 1 was really identifiable by the bacterial communities living on their floors. If I took a swab of their floor, I could tell you exactly that that family lived in that home. This is work done by Simon Lax, one of our really good graduate students. We also know that as soon as a family moved into a new house, their microbiological signature, the bacteria they exuded, basically swamped any other signal in that home. Within 24 hours, I could identify a family by swabbing the microbiota on the kitchen counter or on their light switch. It was transmitted very rapidly. This even occurred in hotel rooms. So within 24 hours, even less actually, now we actually know it's 5 hours. Within 5 hours, the microbiome of your hotel room looks like the microbiome of your home. Of course, the microbiome of your home is made up by whoever lives there. I recently just took my family to China, and so the whole family was stuck inside one room. And that room started, after 24 hours, to look like our home, back at home. But if it's just me traveling, I'm just the only one in the hotel room, then it looks like my den, the place where the kids aren't allowed because they touch my guitars and they hurt them. Very, very sensitive issue. But the family leaves its fingerprint on the home in unique ways. And we can use this. It's an example where a young couple, here in blue and red, The dots are microbial communities. The closer the dots, the more similar those communities are. The further away the dots, the more different they are. So these are samples of their, all the human samples of their hands, their feet, and their noses. We could tell from these samples that the young couple were physically interacting. They were having a relationship. One member of that young couple is in the audience tonight. I can see you. And we knew that you could tell that they were having a physical interaction. You could tell that neither of them were physically interacting with the lodger, not to any substantial effect. But you could also tell that the lodger and the young couple lived together because they were more microbiologically similar than they were to anyone else in the study. So these are adults that came together later in life, and their microbial similarity is increasing because of their shared occupancy of a space. In fact, right now, all of you are becoming about 0.00001% more microbiologically similar over the next hour because you're living in a shared space. So you're increasing your microbial sharing, and the closer you are, the more microbial sharing you have. And we are just starting to understand what this has. I want to go on one very brief trajectory, little tangent. We know that apes, when they groom each other, if they groom each other, the more they groom each other, the more microbiologically similar they are. And we think social interactions like grooming, like kissing, like holding hands, like hugging, may have evolved to actually increase our microbial sharing, so that good bacteria that defended against pathogens from the environment could be shared among your troop. And that the other, that sense of that's the other tribe and we don't go near them, that evolved to stay away from another potential source of pathogens that could infiltrate your tribe. So I'm maybe pushing the limits of your credibility right now, but my credibility. But I think social behavior and social evolution within human societies and ape societies may have been driven by bacterial interactions. And I think that's fundamental. We can monitor and map the microbial interactions. So this is actually my family. We have our feet, our noses, and our hands, and we can tell that the microbiome of our feet interacts with the floor, and we pick up a lot of microbes from the floor and our feet. And kitchen light switches, doorknobs, and counters all interact with our hands. And we can track and monitor the directionality of that microbial flow. We have a map now. Once you have a map, you can manipulate the space. You can understand it better. This is my son. He was a microbial sampling wizard, and he loves doing it now. He's a little scientist. If you can get your kids to do it well. But we know that those microbial communities on his feet were transmitting between his feet and the floor, and then back into my feet, and then over to his mother's feet, and then back into our bodies as we touched our feet. So there's a huge amount of directionality and movement in the microbiome. What's even more interesting, and this is Jared Hampton Marcel's house. You can ask him about his dog obsession later. But Jared has 3 big dogs, and his house was very complex. The dogs increased the complexity of interactions and dynamic movement of microbes around the house in what we believe is a very healthy and good way. It's because dogs are awesome. This is my dog. As soon as we got those results, we went and rescued him from a kill shelter in Kentucky. He had to drive down there and pick the little dog up. He was a little puppy. Who could kill that dog? But fantastic transmitter of microbes. They increase microbial sharing, and we believe, or we hypothesize, that significantly improves our health in a very realistic way. So we're very excited about that. We also know from work done by Susan Lynch that bacteria from dogs can reduce the burden of allergies. This is somewhat paradoxical for most indoor air and epidemiological experts and immunological experts, because they expect, dogs trigger allergies, they make you sick, right? Well, only if you're dysbiotic to begin with. If you have an overactive immune system, then their fur can trigger an immune response. But if you're brought up with a dog, then certain bacteria like lactobacillus that can get inside your body from that dog can significantly reduce the likelihood of having an asthmatic attack in a mouse model. Everything's in a mouse model. Very hard to prove this in humans, but we're starting to explore clinical trials right now. So we are moving out of homes, out of dogs, and maybe moving into hospital environments. We've trained our practices on this. We now want to explore what happens not when you're healthy and living with a home, but when you have to be in a place where you're going to be sick. You're going to have a dysbiosis with your body and with your microbes. Your symbiotic relationship with your body is broken down. You're in hospital. And so this is the new Center for Care and Discovery at the University of Chicago. It's a very expensive building, fully isolated. We looked at the top 2 floors up here, which are patient rooms, beautiful views. If you have to go to hospital, it's got a great view. And we looked at the bacterial community every day from multiple rooms, multiple places, the patients, the doctors, the nurses, the air, the water, the surfaces. And we looked at the bacterial communities, the viral communities, the fungal communities as they changed over time. We saw, for example, that the bacterial communities were very low in diversity, not many species there, when the hospital was non-operational. We got really lucky. We got to go into this hospital and look at it before all the sick people and doctors moved in. As soon as these sick people and doctors moved in, the diversity jumped up. The number of species of bacteria we encountered was significantly increased. That difference, all human bacteria. Many different types of bacteria, and this is not uncommon. I said you're all shedding bacteria right now, and when you're in an occupied space, if you stick a sensor in the middle of this room, you'll see a lot of bacteria floating around. Just as you saw in that little cartoon video, we see all the microbes around us. We can't see them, they're too small, but they're all here. And they are outnumbering us trillions to 1. So that occupied space leads to a massive increase in microbial distribution. And we can track the different species that are there. For example, in patients, we often find a lot of staphylococcus. There are some pathogenic staphylococcus, but most staphylococcus are entirely harmless. We see a lot of Corynebacterium, which is a very common bacteria, but it tends to start to increase on skin if the patient has a dysbiosis, a breakdown in their relationship. On staff, these orange dots, we saw one staff member that had a lot of staphylococcus. On the whole, most of the staff had healthy, normal, high-diversity microbiomes. And so using this information, we can start to track healthcare-associated infections, antibiotic resistance cassettes, understand the development of this ecosystem. Because no matter what we do to our built environment, it is an ecosystem. And bacteria and viruses and fungi will find a way to live. Just like Jurassic Park without the dinosaurs. They will find a way. And if they find a way, we better be sure that we can control that way to make sure it's beneficial for us. Because if we're not doing that, we're missing a trick. So I want to make sure that we put microbes into our patient records. Make it an intricate association. There's one easy way to do that. That's every time you do what you do in the morning, you could have a little sensor in there, and we're developing these at Argonne National Laboratory with Peter Beckman, Charlie Catlett, and groups over in the MCS building, Maths and Computing Sciences. These are brilliant engineers and technicians and computer scientists. Developing a way to relay the microbial community inside your guts every day up to an iPhone. So say you're sitting there in the morning and I use something called Fitbit to monitor my steps. And I look at it sometimes in the morning. Well, maybe it's telling me now that you haven't got quite the right microbial diversity or species inside your gut. You really should eat more blueberries this morning for breakfast on your cereal, or maybe have some bacon. I would program mine to say have bacon every morning, of course, but maybe you could do that. And so every single day you could fine-tune your microbial life and then fine-tune your health. And you could keep track of it as a metric of your health. In a way that, unlike any other metric we have, is dynamic enough to record so much information about your body. Here, for example, these are 2 individuals, 2 men. He has a lot of Bacteroidetes, the blue bacteria. This guy has a lot of Firmicutes, the purple bacteria. As I said, they're microbiologically different. Each one of you is microbiologically unique. We have 2 instances here. I'll blow it up a bit. Here, this is over the course of a year, so we looked at their stool every day for a year. This is Eric Alm and Lawrence David from MIT. Here, Eric Alm ate a dodgy omelette, I think it was, and he got salmonella poisoning. His doctor told him he had norovirus, didn't take any antibiotics, stayed at home, got better, but it turned out from his microbiome profile he actually had salmonella. And his microbiome got back to normal, but he could track that, he could identify it. If he'd been able to do that in the morning, first thing, he could have gone and taken an antibiotic, or maybe just decided, I'm not going to do that, I'm going to see how this works out, but he'd have more information to go on than the blind guess of a doctor. A blind guess is a bit harsh, I don't mean that. Maybe educated thought process. This is an interesting one. This isn't an infection. Lawrence David went and lived with his wife, his new wife in Bangkok, Thailand, for about 6 weeks, and it changed his microbiome. He was breathing different air, drinking different water, having a different community, and it changed his community profile. Using a crowdfunding program that we started up about 2 years ago, we're starting to increase our understanding of the microbial world by getting the public to help fund this science. This is because it's very difficult to get discovery-based science, where we're actually trying to develop a database funded by the government. So we got interested people who are willing to do it and charged them $99 and got their microbiome sequenced. And we've raised about $800,000 so far using this mechanism, so much so that we've now released it in Britain as well. So we're going to do British gut and Australian guts opening up very soon as well. So we're going international. And using this information, this is Michael Pollan's, a New York Times reporter we did, we can start to tell how similar that person is to the rest of the community and how their diet or gender or their body mass index, what role that might be playing in shaping that microbiome and how it might be indicative of that response. We can even look at individual families and track their microbiome over time and see how different species of organisms are more associated with different members of the family, and use that information to understand their health. In this case, the son had crippling allergies, and it turned out he had a very different microbiome. So this is work done by Nasir Sangwan in my lab and Simon Lax. And he had a different microbiome that potentially was leading to the triggering of his allergic reactions. So when they suggested doing this, I thought, well, you know, I could just talk to you, which I have done for probably too long, I apologize. Or I could do a live demonstration of what I think is the state of the art of the power of this technique. And it may not be so impressive, but I asked Peter, you've met Peter actually already, there's Peter down there, and Eric, there's Eric down there, to provide some samples, intimate samples maybe, that we could use to sequence their microbiome and see how they compared to the rest of America. It's a little bit tongue-in-cheek, nobody take offense. I do want a job. This is a lot of work done by Nasir Sangwan in my lab, who's brilliant at doing bacterial assembly. So here's Peter versus Eric. These are all bacterial sequences that we found, genomic sequences from the bacteria in their guts. We found some interesting little components. We took 2 samples, 2 from Peter and 2 from Eric. We compared how similar they were. Eric's samples were actually a little bit more variable. So his 2 samples were more different than Peter's were. But Peter was more diverse. He had a higher microbial diversity in his gut. And the variability in Eric's gut, and this is a certain amount of supposition because he's an N of 1, we think might be due to, haven't told us anything, so I could be completely wrong. This is like doing clairvoyance. Is there a John in the... it looks like he might be eating more plants. And if he's eating more plants, and by that I mean vegetables and fruits and salads, he might be creating more fiber in his gut, and that leads to compartmentalization, which a lot of the work we do in soils with Dion Antonopoulos and Sarah O'Brien at Argonne looks at compartmentalization of microbes in soils. And we think that increases the variability of the microbes. They can live in different compartmentalization. So, Eric, you might have, I don't know, slightly more diverse stool. We also saw a very, very small amount of a really interesting bug in Peter's guts called, I've forgotten the name of it now, that's bad. Nasir, what was that bug called? There we are, Akkermansia municiphilia. That's why I have Nasir around. I can't remember all the Latin names. So Akkermansia municiphilia is negatively correlated with obesity. If you have this bug in your guts, you're less likely to become genetically obese in a system. But Peter, you did have very little of that, so don't get too excited. But it's interesting. We're starting to look at Akkermansia as a potential probiotic to treat people with to reduce the likelihood of obese development. The reason we think you've got more plant-eating capabilities is you have more of this Ruminococcus bromii, and you have more of this Alastipes sharhai, and this Faecalibacterium pranalsii. All these guys are really good at fermenting complex plant carbohydrates. Okay, you see some of them in Peter's gut, but not so many, suggesting that he's eating less of those basically nutrients that feed these bacteria. And so this fermentation, these fermentating bacteria potentially could be indicative of a plant-eating habitat. We compared Peter and Eric to the American gut population, the 4,000 or so people that we've sequenced in American gut, and we said, how similar are people to either, Peter or Eric? And how similar are you compared to that? All of the dots here are more similar to Peter's microbiome, and all the dots here, all the people here are more similar to Eric, okay? So by just comparing, using you two as the standard, we could divide this group into two. The ones that are yellow are really similar to you. So you have a significant similarity to those populations. The ones in purple are less similar. And we asked, what defines these two groups of people? Why are they different? Well, don't take offense. 82% of people with your microbiome, Peter, are exercise very rarely. That's quite an amazing statistic. Doesn't mean you don't. You might run every day. I don't know. And this is a limitation of our technology. It doesn't mean the bacteria make you run less. But they may do. They may change your behavior. We don't know. But we're trying to understand how we can use this as a biomarker for lifestyle. Only 45% of people with a microbiome like Eric's run regularly, but that's a significant relationship. So we also know that 37% of people with a microbiome like Peter's take probiotics regularly. And we didn't exactly find any specific probiotics in your gut. Do you take probiotics? No. Do you eat any cheeses or... Do you eat cheeses? That's right. Yes, that's what I meant, cheeses. 45% of people, 42% of people with a microbiome like Eric's take vitamins. Do you take any vitamins, Eric? Yeah, see, I was right. Yeah. I'll take anything I can get right now. Both of you seem to sleep very well. You both fall within a microbiome that has at least 6 hours of sleep a day. You don't have a significantly disrupted sleep pattern, although I find it hard to believe for the provost of the University of Chicago and the director of Argonne National Laboratory. If you're sleeping, why am I not sleeping? But you are sleeping like babies. This is a good thing. 53% of people with a microbiome like Peter's tend to eat very little plants and more meat. So you're a powerful carnivore and powerful man. I need a raise. 61% of people in a microbiome like Eric's tend to eat 13 plant species or more a week, and more herbivorous. Maybe more like a tortoise. That's the end of my career. So we are very interested in how we can translate this kind of information and make it useful for our populations. We are a human species now living in a very constructed environment, what we call the built environment. And these cities have been created for one purpose. One, to give us housing. Two, they've been created to be microbiologically unfavorable. The environment in these spaces has been designed to kill most bacteria or to be unfavorable to their development. They're very dry environments, very nutrient poor. And we are tracking the microbial communities that are evolving in this environment to see how that ecosystem, it's a very harsh world like Mars, how it's shaping the microbial ecosystem of those spaces, how it's shaping their evolution, their metabolism, their functionality. And we're looking at this space in the context not just of a building, but of an entire city, and using information that we can glean from what's happening inside these spaces to maybe design better cities, better towns, better rural communities that are less shaped by the microbiome and disease and more enhanced by the microbiome. And to do this, we're developing these sensor technologies that we can use to examine pathogens, examine that microbial dysbiosis, that microbial imbalance, look at allergens and pollution so we can influence policy in all kinds of environments, in air, in soil, in water, in human bodies. And if we can do this, we can have a significant impact, a translational impact upon our life. And that's what we're all about, right? We want to take science and make the world better for us. Well, at least that's what I'm about. Everyone else can do what they want. But I want to make the world better. And hopefully, by looking at the microbiome in this way, we can. So I'll leave it there. Thank you very much.
So thank you, Jack. We only have time for a few questions, so if you'd like to ask a question, there'll be people working their way around the crowd. Just raise your hand, and if you would, please hold the microphone close to your mouth so that people can actually hear you.
Anyone brave enough to ask the first question? Eric.
Hey. Hi there.
Oh, no, you got trumped. Where are you? Where are you? Where are you? Oh, thank you.
Thank you, brother. My wife and I have been vegetarians for 40 years, ever since we were 64. Has our biome changed so that meat would be injurious to us at this point?
We have no evidence to suggest that. In fact, people that eat only vegetables tend to have a slightly more diverse microbiome, because even if you wash your vegetables, there are lots of bacteria living inside the plants, and you take those bacteria into your bodies. Those bacteria originate in the soil, so they get from the soil into the plants, and the plants into your bodies. And when you eat those plants, they're packaged nicely inside the plant, so they get past your stomach and they get into your gut. So vegetarians tend to have a slightly more richer, diverse gut, maybe more adaptable. That's circumstantial. We have no evidence of that, but it's, I doubt eating meat again would have any significant impact upon your health. If you want a bacon sandwich, maybe. Yes, ma'am. Oh, that one's got a microphone.
I've suffered from fevers for 3.5 years, and on many occasions been septic with different bacteria. Do you have any recommendations or education that might help me?
I'm not a doctor, I'm a microbial ecologist. I would say we could do an experiment on you and find out what happened, but then I'd probably get in trouble with the ethics committee. It's complex. You know, one of the things about Western medicine is we want a one-size-fits-all solution that I can just roll out and charge $100 for, and everyone will get better. That's not the way it's not the way your body works. The microbial world inside you is quite unique. If I wanted to understand your condition better, I'd want to know about your daily routine, about your lifestyle, maybe even live with you, not suggesting this, but for a period of time, so I can get to know your routine, see your habits, understand your diet. We trick ourselves and we don't tell people quite the truth about our lifestyle. Quite often observing somebody in their home can be a far more, a better, more accurate perspective on their lives. So that's where we're trying to get at, personalized medicine, that it really can treat your condition, not just the ailment which you're suffering from. Yes?
Hi, we have 2 sons, and the 26-year-old has a lot of trouble getting above 140 pounds. The 19-year-old has a lot of trouble keeping under 320 pounds.
Right.
At some point, is there a likelihood that there could be like a poop transplant that might help them both out?
Well, fecal microbiome transplants are a very present medical treatment. The Chinese in about 500 BC called it yellow soup. So they would feed stool from one healthy person to a sick person in order to cure disease. There was a Swiss doctor who treated Hitler with microbiome feeding. So he fed him feces from a healthy person. I think this is just a great way to make Hitler eat. But anyway, make a bad person do bad things with what they shouldn't be doing. But recently, we've been really, since the 1960s, we've started to investigate this, but very recently we've uncovered ways to use fecal microbiome transplant to treat a myriad of diseases. It's a bit of a shotgun approach, though, pardon the expression. But placing a new microbiome up inside your colon is a way of just basically saying, we don't know what the problem is, but we know there's something wrong there. If we swap it out, hopefully it will fix the problem. It works in mice. It works in humans with conditions like C. difficile, infections which are pernicious and terrible. And we can treat those conditions. But it doesn't mean it's a panacea that can fix everything. We need to understand the mechanisms. And again, like I said previously, I'd like to understand your son's lifestyles in intricate detail in order to be able to provide any appropriate experimental study design to understand what was going on. But it's possible that we could look at probiotics or specific types of organisms, not the things you buy in Whole Foods, which are 99% of which have very little influence upon our health. But the actual specific organisms, like I've talked about today, that could have an effect on that.
So time for one more question.
I'll grab your question afterwards.
So I was going to ask a question more related to the science. So if there are 6 billion people on Earth, so that's what, 2 pounds per...
7.2 billion.
So it's about 7 million tons of these microbes in humans. Yeah. So it's a 2-part question. The first is, how does that relate to the total mass on Earth? And how much does the human you know, presumably when you ingest these microbes, your body does some shaping of the distribution of those microbes.
Absolutely.
How much does it shape I mean, what fraction is it of the total, and how much can it shape actually the environment around you, aside from the very local effect you described in hospitals and
Sure.
kitchens?
There are a nonillion bacterial cells on Earth. There's literally kilograms of bacteria. They outnumber the stars in the known universe. Unless you're really into astrology, bacteria are far more fundamentally important for your health than astronomy. So we should shut down that whole space program and just give the money to me. It's you know, physics, stop it. Biology is everything. So fundamentally, already we have an issue. We have tons and tons of bacteria in the world, but our body doesn't stop at this thin integument, right? We are intricately linked to that environment. Every breath you take, every move you make, I'll be watching you. We know that relationship exists, and we're taking those bacteria in and we're shaping them on the outside. Your body does a lot of shaping. Using a project called the Earth Microbiome Project, we've identified now about 12 million bacterial species. And there are only maybe about 4,000 or 5,000 of those that regularly colonize human beings. So that's already an enormous step down from 12 million down to 4,000 or 5,000. That selection process that your body undertakes is quite distinct. It's because it's a weird ecosystem. It's anaerobic. It's warm. It's moist. It's a very different ecosystem to what bacteria experience in the majority of the rest of the world. And so we need to understand that ecosystem response so we can really fundamentally track how those microbes are affecting not just our health, but the health of our environments as well.
So thanks, everybody. If we could hear one more time for Jack Gilbert.
Sure.